In this video, we will be describing the histology that you will view during exercise 4. Please watch this pre-lab video before attending lab this week. In order to follow along with the video, be sure to have your lab manual open to page 7 where it says Exercise 4 Histology, and to have the Histology Lab Atlas that you received from your instructor handy. You may make notes in both of these items as I go through the description of each of the tissues. In the first part of the video, we will describe the many types of epithelial tissue, also collectively referred to as epithelium. We will then move on to the different tissues that are referred to as connective tissues. This is where we will stop for this lab, and we will continue our discussion of histology as needed through different body systems. To begin with, we look at what is called simple squamous epithelium. Simple refers to the fact that this kind of tissue has only one layer of individual epithelial cells. Squamous refers to the fact that these cells are flat and pancake-shaped. When you look at this image, such as what you would see after scraping your cheek and smearing it onto a slide, you can see the individual cells side by side. Here we've outlined a cell for you. This line would essentially represent the plasma membrane, and this dark dot in the center would represent the nucleus of the cell. While it is not labeled, you should know that the space in between the plasma membrane or the cell membrane and the nucleus is referred to as the cytoplasm. You can see a lot of individual cells here side by side. They are not outlined like the one that we have colored in for you. Many of their borders overlap, so it's kind of a messy look, but you'll know every time you see a dark dot that that's the nucleus of a single cell. Simple cuboidal epithelium is another type of tissue. You will see still a single layer of cells, but what we're looking for now are the single layer of cells that form what are called here kidney tubules. You see a series of these tubules. A tubule like this has a hollow space. We refer to that as a lumen. This is true of any hollow organ, and in this case you can see substance inside the lumen. Since these are from the kidneys, the substance in the lumen in this case would be the early forms of urine. While we're looking at epithelium itself, we actually have a slide of an organ. This is an organ, the kidney, and an organ itself is made up of 2 or more tissue types. So we have pointed out that in between the tubules you do have types of connective tissue, and so you see it present, but we are just going to focus in this video on the cuboidal epithelium that forms the tubules itself. This image is at 100x total magnification, and so the next image shows you a zoomed in version, showing you one single kidney tubule. So again, this tubule is formed of a single layer of cells. That's why it's called simple. And the cells have a different shape than what we saw in the previous slide. These are now a more box-like type cell. These are always referred to as cuboidal cells. And you can see again the cellular anatomy labeled here in the picture for you. This is the plasma or the cell membrane. This is the nucleus. And what makes this epithelium is that the cells are bordered side by side. And there are no spaces in between. They are close together. That way the tubule doesn't leak. You also note that because it's epithelium, one side of the cell here is going to be bordered to connective tissue of some sort, while the other side is exposed to whatever is in the lumen. Great representation of epithelium right there. We move to another type of tissue called simple columnar epithelium. Again, this is from a hollow digestive organ. This is likely part of the intestine. And an organ itself, again being composed of 2 or more different types of tissue, you'll see here in all of this space is going to be a connective tissue, and there's probably smooth muscle and adipose associated with it. But since we're studying epithelium at this point in time, we want to focus on the part of the organ that faces the lumen. So again, right here, this hollow space. We're going to zoom in right here to take a look at what is known as the apical surface of the epithelium. Those little finger-like projections here are jutting out from the connected part of this hollow organ and into the lumen. So here's the hollow space. The cells themselves are still a single layer. So if you run your finger along the cells, starting here with the nuclei, you see they line up side by side in one row. All of these cells are adhered by their basement or their basal layer onto a basement membrane. And then this pulp that's between the 2 layers of cells here and here is actually made up of connective tissue. Because this is a part of the organ called the mucosa, we refer to this connective tissue region as the lamina propria. Going back to describing the epithelium itself, not only is this a single layer of cells, but these cells themselves have a definite shape. If you trace the individual cell, you see it has a column shape. As a result of that, we refer to this tissue as simple columnar epithelium. There you go, all the way around. Now, it is quite common that in regions of the intestine and the respiratory system that you see another type of cell called a goblet cell. The goblet cells are represented here by a space. Now, a goblet cell itself looks like a goblet. You'd see the open top of the cup right here, and you'd have the stand actually attaching down here to the connective tissue. So same thing right here. You can see the open space where you would fill it with fluid, and there's the stand or the base of the goblet itself. Goblet cells produce mucus, and that mucus is secreted from the cell here into the lumen. It will coat the level of the epithelium that is apical. That way, anything inside of this hollow organ slides through it more easily. The next type of epithelium is called pseudostratified, ciliated columnar epithelium. We're once again looking at a hollow organ, but in this case it is the trachea, made up of multiple types of tissue. You can see adipose tissue here at the base, hyaline cartilage, followed by loose connective tissue in this region. It includes the lamina propria. And then you see your epithelium. So we're going to focus on the epithelium itself. I will note once again that we know it is epithelium for a number of reasons, including which is that it faces the lumen, the hollow space of the trachea. So if we zoom in here from 100x to 400x, we again see our thick band layer of the epithelium. To begin with, the word columnar should sound familiar. These cells are column-like in appearance. It's a little more difficult to tell that column shape than it was in simple columnar epithelium. So we've drawn this column-like shape here to show you the direction that these cells all run. They are anchored here at the basal lamina towards the lamina propria, and their apical surface sticks out here into the lumen. What is unique about this tissue is that it has little cellular projections called cilia, hence the name ciliated. These cilia beat and move whatever is on the surface of the cells in a similar direction. So in this case, because it's the trachea, these cilia work together to propel mucus and debris up through the respiratory tract and then back down to the esophagus. Going back to the cells themselves, even though they are columnar, there is a difference here in the nuclei than what you saw with simple columnar epithelium. With simple columnar, the nuclei were all in one line, but here with pseudostratified ciliated, you see the nuclei are actually juxtaposed to one another and in different layers, it appears. This is a trick of the eye because this is still a single layer of cells, even though the nuclei are at different heights. All the cells are anchored here to the basal lamina and the basement membrane, and some are tall, so their nuclei appear higher up, but some are short, and so their nuclei sit a little lower down. Because it is falsely stratified, we call it pseudostratified. Transitional epithelium is the first true stratified tissue that we look at. So to begin with, looking at this particular organ, which is the urinary bladder, that's one of the most common places we'll talk about transitional epithelium. You can see lamina propria here at the base, so made up of connective tissue, and the epithelium is in this region from top to bottom. Here's the basal layer of the epithelium, and here's the apical layer of the epithelium touching the lumen. There is more than one layer of cell here, and you can judge that by the fact that the nuclei do stack on top of one another. And if you look real close, you can usually see cell borders. So here is a plasma membrane of one cell with its nucleus. Here's another plasma membrane with its nucleus, and it sits on top of this one right here. So it's truly stratified, not pseudostratified. Transitional epithelium is unique because the cells at the very top appear globular and cuboidal whenever the urinary bladder is empty. But when it fills and this apical surface begins to stretch, the cells take on a more squamous appearance and they flatten out. So it transitions from one shape to another. The next type of stratified tissue we look at is nonkeratinized stratified squamous epithelium. So again, you can see in this image the lamina propria made of loose connective tissue sitting below or basal to the epithelium, and the epithelium itself is multi-layered, reaching all the way to the lumen. At the very base here of this section of epithelium, you can see the cells are larger. So these are cuboidal epithelium down here at the very bottom. But as you move to the more apical surface, you see the cells begin to flatten out. This is most clear when you look at the nuclei. You can see the nuclei here are very flattened as opposed to nice rounded nuclei here. Now these cells are squamous cells at the very top. So despite the fact that there are two shapes of cells found in this tissue type, we name the tissue overall for what kind of cell structure lies in the most apical regions. So it's more than one layer, it is stratified, and it is squamous epithelium. The term non-keratinized describes the cells that are in the epithelium. It indicates that they lack a protein called keratin, a protein that tends to harden and protect these cells. Non-keratinized stratified squamous epithelium is typically found lining tubules inside the body or cavities inside the body where you have production of mucus and fluids. This is different from what we see in keratinized stratified squamous epithelium. Keratinized is going to be a type of tissue which is found on the outside of the body, which is exposed to the elements. The skin is the example that we look at here of keratinized stratified squamous epithelium. So the skin itself is divided into three primary layers. The hypodermis, not shown, is composed primarily of adipose tissue. The dermis, the next deepest layer, is actually divided into two regions, the bottom reticular layer here, and then running at about this level here, starting here and running up and curving around, that begins the papillary layer of the dermis right here. These are both connective tissue. The epidermis itself is where we find our epithelium. So we'll focus in on that, moving up to 400x magnification. Again, here and below, you see the papillary layer of the dermis. So let me run the pointer along the border between dermis and epidermis. That's the separation between the two. The epidermis itself is all going to be composed of epithelial cells. You can see here, like we saw in the non-keratinized stratified squamous epithelium, that the cells start out in one shape at the very bottom, which is cuboidal, and as they move closer to the lumen in this apical region, they begin to flatten out and become squamous. That's why the tissue is named for squamous. Keratinized can be most indicated by the appearance of the cells at the very top. Whenever the protein keratin becomes more numerous within the cell, that's going to represent that the cell is hardening more or less to become protective in nature. There are other physiologic changes we will talk about that are associated with this that cause this layer of skin to essentially be dead. We call this layer the stratum corneum. The layer right below that is this nice dark line of cells. It's a single layer. We call that the stratum granulosum. Below that dark layer, you can see a lighter pink region where the nuclei are nice and round. It follows all the way through here. That's multiple layers. That's referred to as the stratum spinosum. And the final layer here is called the stratum basal. It's a single layer of cells. They do tend to have the darkest bands of nuclei along the bottom. You should know all four layers of this particular tissue and the epidermis. Keep in mind that this is an example of the stratified squamous epithelium known in what is called thin skin. The other kind of skin that we look at is called thick skin. Thick skin is found in the soles of the feet and the palms of the hands. If we examine a tissue slide of keratinized stratified squamous epithelium from thick skin, we see the other characteristics of skin that we saw previously. So to begin with, you see a hypodermis at the very base here, made up mostly of adipose, a nice thick layer of the dermis divided into two layers, first reticular, then the papillary layer. Papillary layer is most notably named right here for the fact that it gives off finger-like projections called papilla. You can see those indenting their way here into the epidermis. And then, of course, you see the darker cells making up the epidermis. When we begin to magnify those layers here, we start to see some familiar terms. We still see a single layer of the stratum basal here at the very base of the epidermis. We see another 5 to 6 layer region here called the stratum spinosum, followed by a dark layer called the stratum granulosum, and right above that, a light layer of translucent cells known as the stratum lucidum. This is only present in thick skin. Then we finally see a stratum corneum here at the very top. Now, you will notice in the images that sometimes these cells of the stratum corneum begin to separate because of the keratinization, so they don't stick together as well. So there are certain spaces that begin to appear, and you start to see these dead cells begin to flake off. Don't let that confuse you. Start to use your stratum granulosum as a key point for representation. Look for that dark line in order to indicate the location of stratum lucidum to you. That's the end of epithelial tissue. We now move into areolar connective tissue. Areolar connective tissue is essentially an example of the different types of components that you should see in most types of connective tissue. So to begin with, connective tissue is pretty different from epithelium in the fact that the cells are more spread apart. So you can see the nucleus of a cell here and a lot of space followed by the nucleus of another cell. Space followed by the nucleus of another cell. The particular cells in this image are known as fibroblasts, and these are the nuclei of the fibroblasts. They're so named because they produce or they secrete the protein fibers that make up the rest of the connective tissue. These connective tissue fibers are the second component of connective tissue, and actually they are more predominant than cells in some cases. In this image, you can see black fibers, which are known as elastic fibers, and pink fibers, which are known as collagen fibers. This is another slide. It's very similar to the one we just saw. It is called areolar connective tissue with mast cells. So these are two separate slides that you'll use in lab. Once again, you can start to see fibroblasts. One thing you'll note right here, as you saw in the image previously, is these nuclei tend to be somewhat flattened. Right? So flattened fibroblast nuclei, making the many fibers. You see black elastic fibers and then your pink collagen fibers. But you see a second kind of cell right here called a mast cell. A mast cell, I can outline for you right here, actually has a plasma membrane that wraps around in this region right here. And so right in the middle you see the nucleus and you see the plasma membrane, and then you see these tiny little dots. The tiny dots are actually granules which exist within the mast cell, and they give it its function. There are other types of cells that you'll see within areolar connective tissue, but these are the only two that you must identify in these particular slides. We move on then to adipose connective tissue. It is still a type of loose connective tissue. These are individual adipocytes, that's the cell name. They're also called fat cells, but this is a more scientific term. And these cells are recognizable by their giant inclusions called oil droplets. The oil droplet within the cell takes up so much space that the nucleus, which is normally in the center of the rest of your cells, gets pressed up against the plasma membrane here on the side and flattened. So they tend to take on an appearance of eyeballs, which are looking at one another. So here is the iris of one looking at the second eyeball. Here's another eyeball, if you will, an adipocyte with its iris looking out this way. So very recognizable tissue. Reticular connective tissue, also quite recognizable from the vast array of reticular fibers that you would see running all through it. Reticular fibers are the third type of protein fiber that we see in these examples. And reticular fibers tend to be pretty tough, and they provide a lot of structure for an organ made with reticular connective tissue. You will still see fibroblasts, notated here by dark nuclei, and these fibroblasts are producing these reticular fibers. Dense regular connective tissue has two other names that we examine. It's also called white fibrous connective tissue, or it's called collagenous connective tissue. Here you again see fibroblasts, so they are the tiny dark nuclei of the fibroblasts. Those fibroblasts have secreted pink collagen fibers. What is unique about these pink collagen fibers is that they lie in parallel patterns to one another. And so they give you this almost wave approach as you examine the tissue. This is what makes this tissue regular. All the collagen fibers are running in the same direction, parallel to one another. It's referred to as dense because there's very little space between the individual collagen fibers. They are densely packed. We now move on to three types of cartilage. Cartilage is going to have a few similar characteristics. Two of the types of cartilage that we look at will have an outer border called perichondrium. That literally means around the cartilage. It is a very thick layer on the outside right here. You will notice that it picks up more stain than the rest of the cartilage, and that it's going to have fewer of the very representative cells that we see. We refer to these cells as chondrocytes, and in this image they are actually sitting in pockets called lacunae. The space in between these lacunae are actually called matrix. It's called the matrix, and it is collagen rich. That's the reason that you have a slight pink appearance with this tissue. When we zoom in, we can see these chondrocytes and lacunae quite clearly. So there's your pocket, your lacuna, and the individual nucleus of the cell. That's your chondrocyte. Elastic cartilage connective tissue looks darker, primarily because there's a different type of fiber here in the matrix. While there was pink collagen with hyaline, here in the elastic cartilage connective tissue, the dark purple and blue is due to the presence of more elastic fibers. You can still see perichondrium here on either side, and you can still see the chondrocytes and lacunae right here within the matrix. You can see in this zoomed in image much better, there's a single lacuna and the nucleus of a chondrocyte. Moving to fibrocartilage, this is the third and final cartilage tissue we look at. This does not represent perichondrium. It is the only kind of the three that does not have that perichondrial border. In this particular image, you're actually looking at what is called the pubic symphysis, made of fibrocartilage. And the pubic symphysis exists between two regions of bone, which you see right here. The image often looks a little bit like a fly. Here are the fly's eyes on either side, and here's a nose right down here in the very middle. So the perichondrium, if it existed, would sit right here, but it is missing. So that's a key point in trying to identify this slide. The fibrocartilage still has the very characteristic appearance of cartilage itself. You will note when you look at it here, okay, this is cartilage, the beginning of the cartilage moving in this direction. Here's the bone moving over in this direction. You will see the chondrocytes and lacunae moving all the way through the fibrocartilage. We also have compact bone connective tissue. This compact bone itself will be compared with spongy bone later on, but for now this is the only type that we are looking at. And it's highly recognizable because of the presence of what are called osteons. And each osteon itself looks like a tree with a core and rings. This can also be called a haversian system. The anatomy of this individual system consists of a central canal. It consists of rings which wrap around that. When these are a part of the osteon itself, they are called concentric lamella. But when they're outside of the osteon, running around the bone as a whole, they're called interstitial lamella between osteons. And as you zoom into this image, you begin to see that the cells themselves run along the pattern formed by an individual lamella. These cells are very similar to cartilage cells in that they are found in pockets called lacunae, and the cells are named for bone. They are called osteocytes. Chondrocytes for cartilage, osteocytes for bone. Branching off of the lacunae are tiny web-like tubules that are called canaliculi. These are responsible for the communication of chemical signals and nutrients between the individual osteocytes. We will pick up with our discussion of muscle tissue later this semester, and so we'll stop our discussion here. Please let us know if you have any questions.